Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine, exploring a novel way to predict survival times in dogs diagnosed with a common and aggressive cancer, multicentric high-grade B-cell lymphoma. This research, conducted by a team of scientists and veterinary professionals across Japan and the United States, utilizes cutting-edge genomic techniques to identify specific markers that can help predict how well a dog with this type of lymphoma might respond to standard chemotherapy treatment. Multicentric high-grade lymphoma is, unfortunately, the most frequently diagnosed form of lymphoma in our canine companions, accounting for a significant majority of all lymphoma cases. Within this broad category, the majority of these cancers originate from B cells, a crucial component of the immune system. While significant advancements have been made in treating this disease, primarily through chemotherapy regimens based on a combination of drugs known as ChOP, the outcomes for individual dogs can vary considerably. Some dogs experience long periods of remission, while others may relapse quickly, highlighting the heterogeneous nature of this cancer and the challenges in predicting individual prognosis. Veterinary oncologists currently rely on a variety of factors to estimate a dog's prognosis, including the stage of the cancer, the dog's overall health, and the presence of certain clinical signs like anemia. More advanced classifications, involving detailed microscopic analysis of tumor samples and specialized molecular tests, have also been explored to gain a deeper understanding of the factors influencing survival. However, many of these advanced techniques require invasive biopsies and may not be readily available or practical in everyday clinical settings. Conversely, while clinical data offers a more immediate assessment, the interpretation of certain criteria, such as the classification of clinical substages, can sometimes be subjective and may vary between different veterinarians. This underscores the need for more objective and easily accessible tools to help predict the course of this disease in individual dogs. In recent years, the field of epigenetics has emerged as a powerful area of research, offering new insights into the mechanisms that control gene expression without altering the underlying DNA sequence. One key epigenetic mechanism is DNA methylation, a process involving the addition of a methyl group to a cytosine base in DNA, particularly when it is followed by a guanine base, a sequence often referred to as a cytosine phosphate guanine site. Aberrant DNA methylation has been implicated in the development and progression of various human cancers, including diffuse large B-cell lymphoma, a counterpart to the canine disease studied here. In human lymphoma, abnormal methylation patterns at specific cytosine phosphate guanine sites have shown promise as prognostic indicators. Given these findings in human medicine, researchers have begun to investigate the role of DNA methylation in canine lymphoma. While previous studies in dogs have used advanced sequencing techniques to explore the broader landscape of DNA methylation and its association with prognosis, there's been a need to identify specific cytosine phosphate guanine sites that could serve as reliable and practical prognostic markers for multicentric high-grade B-cell lymphoma in dogs. This current study directly addresses this need by employing a comprehensive, genome-wide analysis of DNA methylation patterns in a cohort of dogs with this aggressive cancer. The overarching objective of this research was to pinpoint specific cytosine phosphate guanine sites that are associated with longer survival times in dogs with multicentric high-grade B-cell lymphoma treated with the standard ChOP chemotherapy protocol. The researchers embarked on this investigation by first enrolling 2 groups of client-owned dogs diagnosed with multicentric high-grade B-cell lymphoma. The first group, referred to as the test group, comprised 24 dogs diagnosed and treated at the Veterinary Medical Center of the University of Tokyo between 2007 and 2010. The diagnosis in these cases was based on clinical findings and a microscopic examination of cells obtained through fine needle aspiration of enlarged lymph nodes. These cases were further classified according to established veterinary guidelines for lymphoma staging. In addition to these lymphoma samples, the researchers also included DNA methylation data from peripheral blood mononuclear cells of 3 healthy beagle dogs from a previous study for comparison. The second group, the validation group, consisted of 100 dogs with naturally occurring multicentric high-grade B-cell lymphoma from various private veterinary hospitals across Japan. In these cases, cytology slides used for diagnosis had been submitted to a diagnostic company. A board-certified veterinary pathologist confirmed the diagnosis and staging of these cases based on clinical findings and the microscopic appearance of the cells. Similar to the test group, All dogs in both cohorts had primary nodal lymphoma, received CHOP-based chemotherapy as their treatment, and had preserved genomic DNA or cytology samples available from the time of diagnosis before treatment. Survival times for all dogs were carefully recorded based on reports from the primary veterinarians and a review of their medical records. To analyze the genome-wide DNA methylation patterns in the test group, the researchers utilized a technique called digital restriction enzyme analysis of methylation, or DREAM for short. This method allows for the quantitative assessment of methylation levels at a large number of cytosine phosphate guanine sites across the entire genome. In the test cohort, this analysis successfully generated a substantial amount of data, with millions of unique DNA sequencing reads for each dog, allowing for the analysis of over 100,000 cytosine phosphate guanine sites per animal. Upon analyzing the methylation data from these vast numbers of cytosine phosphate guanine sites, the researchers focused on those located within cytosine phosphate guanine islands, which are regions of a genome with a high frequency of cytosine phosphate guanine sequences and are often associated with gene regulation. Intriguingly, hierarchical clustering analysis, a statistical method used to group similar data points together, revealed that the 24 dogs with multicentric high-grade B-cell lymphoma in the test group could be separated into 4 distinct groups based on their cytosine phosphate guanine island methylation patterns. When the survival times of these 4 groups were compared, a statistically significant difference was observed, suggesting that these methylation patterns were indeed linked to how long the dogs lived. Further analysis comparing the group with the longest survival time to the combined group of the remaining dogs showed an even stronger statistical significance in survival differences. To pinpoint the specific cytosine phosphate guanine sites responsible for these differences in survival, the researchers performed a comparative analysis between the group with a better prognosis and the group with a more standard prognosis. This analysis identified over 1,000 differentially methylated cytosine phosphate guanine sites, where the level of methylation was significantly different between the two groups. Among these, cytosine phosphate guanine sites located near the genes FAM213A and PHLPP1 stood out as promising candidates. Specifically, these sites showed lower levels of methylation in the group with the better prognosis. To validate these initial findings, the researchers then turned to the validation cohort of 100 dogs. In this larger group, they used a more targeted technique called Bisulfite pyrosequencing to precisely measure the methylation levels at the identified cytosine phosphate guanine sites near the FAM213A gene, referred to as DMCF, and the PHLPP1 gene, referred to as DMCP. After excluding some cases due to their lymphoma type or lack of survival data, the methylation levels of DMCF were successfully measured in a high percentage of the remaining dogs, and the methylation levels of DMCP were similarly well measured. Analysis of the validation cohort revealed that dogs with a methylation level of DMCF below a certain threshold, specifically 40%, had a significantly longer median survival time compared to those with a methylation level above this threshold. While the methylation level of DMCP alone did not show a statistically significant association with survival time in this cohort, an interesting and potentially very important finding emerged when the researchers looked at the combined methylation status of both DMCF and DMCP. Dogs that had both a methylation level of DMCF below 40% and a methylation level of DMCP below 10% exhibited an even more pronouncedly favorable prognosis, with a significantly longer median survival time compared to all other dogs. Further statistical analysis, considering other known prognostic factors such as sex, age, cancer stage, and substage, indicated that on its own, only the DNA methylation status of these cytosine phosphate guanine sites was significantly associated with survival time. However, when multiple factors were considered together, both the DNA methylation status and the stage of the lymphoma were found to be independently and significantly associated with survival. Importantly, there were no significant differences in other reported prognostic factors between the good and standard prognosis groups identified by the methylation analysis. The findings of this study provide compelling evidence that genome-wide DNA methylation analysis can effectively identify prognostic cytosine phosphate guanine sites that can help predict the survival time of dogs with multicentric high-grade B-cell lymphoma treated with CHOP chemotherapy. The subsequent validation of 2 specific cytosine phosphate guanine sites, near the FAM213A and PHLPP1 genes using bisulfite pyrosequencing in a larger cohort of dogs further strengthens these findings. This research builds upon previous work exploring the epigenomic landscape of canine diffuse large B-cell lymphoma, a disease closely related to the multicentric high-grade B-cell lymphoma studied here. While earlier studies identified groups of dogs with different prognoses based on broader methylation profiles, this study goes a step further by pinpointing specific cytosine phosphate guanine sites that appear to be critical in delineating cases with significantly longer survival times. Given the limitations of relying solely on clinical and pathological data for predicting treatment responses in canine lymphoma, the identification of these methylation markers holds significant potential for clinical application. The ability to objectively assess the methylation status of these two cytosine phosphate guanine sites could provide veterinarians with valuable information to share with pet owners, particularly when discussing treatment options and setting realistic expectations for potential outcomes with CHOP chemotherapy. This could be especially helpful in cases where dogs are classified into the favorable prognosis group, as it could provide reassurance to owners who might be hesitant to pursue chemotherapy. Furthermore, as new treatment strategies for canine lymphoma emerge, such as chemoimmunotherapy and virotherapy, these prognostic markers could potentially help identify dogs who are most likely to benefit from these advanced therapies. While genome-wide methylation studies can be costly and time-consuming, the fact that this research has identified specific cytosine phosphate guanine sites that can be analyzed using the more targeted and cost-effective technique of bisulfite pyrosequencing makes these findings more readily translatable to clinical practice. Bisulfite pyrosequencing offers the advantages of lower cost and a faster turnaround time for results compared to more comprehensive genomic analyses. Moreover, the use of DNA, which is more stable and easier to handle than RNA, further enhances the practicality of this approach in routine clinical settings. Importantly, pyrosequencing provides an objective and reproducible assessment of methylation levels, reducing the subjectivity associated with some traditional methods. The researchers acknowledge that the interpretation of pyrosequencing results will require careful consideration. The specific cutoff values identified in this study for defining prognostic groups may need to be validated and potentially adjusted in different populations of dogs, given the known heterogeneity of diffuse large B-cell lymphoma. While this study focused on two key cytosine phosphate guanine sites, the initial genome-wide screening identified a larger number of differentially methylated sites that could potentially contribute to even more accurate prognostic predictions. However, the inclusion of more sites in a clinical test would need to balance the potential for increased prognostic power with the practical considerations of cost, time, and the amount of DNA required. The researchers concluded that the two cytosine phosphate guanine sites identified in this study provide a promising and cost-effective starting point for improving prognostic assessment in canine multicentric high-grade B-cell lymphoma. In conclusion, this groundbreaking study has successfully identified and validated specific differentially methylated cytosine phosphate guanine sites that can predict the treatment response and survival time of dogs with multicentric high-grade B-cell lymphoma undergoing CHOP therapy. These findings highlight the potential of these cytosine phosphate guanine sites as valuable prognostic biomarkers and demonstrate the feasibility of using bisulfite pyrosequencing as a practical clinical test to provide a rapid and objective evaluation of prognosis in dogs with this common and challenging cancer. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.